Welcome, everyone. Thank you for joining us for our presentation titled Multiple Myeloma Screening, Past, Present, and the Future. It is my pleasure to introduce to you Professor Kareem Azab, Assistant Professor, Cancer Biology Division, Department of Radiation Oncology, School of Medicine, Washington University in St. Louis. The research in Professor Azab's lab has a multidisciplinary translational approach involving cancer biology, immunotherapy, tissue engineering, and drug delivery. He has a special interest in tumor biomarkers, tumor microenvironment, drug resistance, and metastasis in hematologic malignancies, particularly in multiple myeloma. He also focuses on the development of ex vivo 3D tissue engineered cancer models for drug development and personalized medicine. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Now, if any questions arise during the presentation, we encourage you to submit them in the Q&A box. Our speaker will address your questions following the presentation. Please join me in welcoming Professor Azab. I will now turn the presentation over to him. Professor Azab, you may now begin your presentation.
So thank you, Christy, for this nice introduction. And I'm honored and happy to be here and giving this presentation. The title of my presentation will be Multiple Myeloma Screening, Past, Present, and Future. For disclosures, the support of this program is provided by Abbott, and the information presented is based on my interpretation of the evidence and the clinical experience. Multiple myeloma is a cancer of plasma cells. And plasma cells are very differentiated B cells. They're the end of differentiation of B cells, and they are responsible for fighting invading exogenous materials like bacteria and viruses by secretion of antibodies to the system. The problem that when these become malignant, they over-occupy our bone marrow and have a lot of side effects and a lot of problems that cause in the end to death. Part of that process is causing low blood counts, because the myeloma cells just push away all the other normal cells in the bone marrow. And we have also a lot of bone destruction. We get a lot of bone fractures in these patients due to bone destruction. And also a lot of infections because we have our immune system suppressed. And finally, a lot of patients die of kidney problems, mainly kidney failure, because of the huge amounts of non-functional antibodies secreted into the blood that the kidney needs to take care of. Currently, if we look at the statistics of multiple myeloma over the last 30 years, we see a small increase of the prevalence of cases. And despite all the new wonderful treatments that we have been seeing in the last 10 years or so, we didn't really see a lot of decrease in the number of deaths, which is a little frustrating, but soon we will probably explain why, or try to explain why. So looking here at the last, it's almost stable for over the last 30 years, which means that there's something fundamental that we don't really understand, or maybe not even in the treatment or in the diagnosis of myeloma. So if we wanted just to look at how multiple myeloma is diagnosed right now, so there are the standard lab tests that like blood counts, blood chemistry, levels and albumin levels. But we also have some specific tests, which are the globulin, the immunoglobulin levels. And these are because, as we said before, the myeloma cells are specialized in secreting immunoglobulins. So the cancer of these cells will start secreting a ton of immunoglobulins into the blood. So we can detect these or fragments of them in the blood, and this will be an indication that myeloma is occurring. Same thing, we can also detect them in the urine. especially the fragments of these antibodies. If we detect them in the urine, then that means that there is a myeloma going on. However, these are more like global tests that look at the entire body at the same time. To go a little more specific and a little more deep into the tissue level, we have imaging approaches like X-ray, PET-CT, MRI, the standard imaging strategies. And these would be more in the organ level, where we see which parts of the body there is involvement of multiple myeloma, which is mainly in the bones around the body. However, these will not tell us any cellular level information. And for this, we take bone marrow biopsies. So we take a bone marrow biopsy or an aspirate, and we examine the aspirate on a cellular level. So we look at the appearance of the cells, the shape of the cells, and how they are arranged in the bone marrow, and we can also do some molecular stuff on them. For example, we do FISH, fluorescent in situ hybridization, which tells us more about the DNA structure and the DNA content of the cells, if they have fragments, if they have multiplications and things like that. We look at immunohistochemistry, which tells us about the cells and how they look like. But these two, the problem that they are more qualitative than quantitative. And to do more quantitative analysis, we have two ways. One is flow cytometry, where we take cells and we stain them with antibodies, and we analyze the bone marrow, including the multiple myeloma cells, for expression of different markers. Then we learn quantitatively about the amount of multiple myeloma and the cellular nature of the multiple myeloma cells. In addition, we can do cytogenetics, mainly next generation sequencing, to learn about the genetics of the multiple myeloma cell clones. And if we want to look at these two, which are pretty powerful, and compare them kind of head to head, so they are both quantitative, they're both sensitive. The next generation sequencing will tell us about the clonogenicity, or which genetic clones are there. So that's really a plus, while flow cytometry cannot do that. However, this one, the next gen sequencing, needs a baseline sample all the time. So to compare it through the progression, while flow cytometry doesn't, which is actually a plus for flow cytometry. Technically, the next generation sequencing is an available service commercially. However, it's not available in most hospitals, and it's not available in most academic institutions. So you need to send that out, to outsource it to companies to do that. While flow cytometry is wide available everywhere, almost in every hematology clinical or research lab, in universities, in hospitals, and everywhere. So that's a plus for flow cytometry. As of complexity, the machines and the bioinformatics needed for next gene sequencing is much more complicated and expensive. For flow cytometry, it's pretty simple. All what you need is standard software. And for the turnaround, which sometimes very important, especially in relapsed patients, for next-gen sequencing, we need about 2 weeks, while the flow cytometry, you get the results in 3-4 hours. That's, again, a huge advantage for flow cytometry. And the cost. The cost per sample for an NGS is about $1,500 per sample, while for flow, it's $300. So there is kind of a clear advantage for flow cytometry over next-gen sequencing. A lot of people would agree with me. Some of people will not, but at least from technical point of view, flow cytometry is easier and more available. So currently, there are 3 markers to identify myeloma cells by flow cytometry, and I'll start from the bottom up. So we start with CD45, which is a pan-leukocyte marker. It just identifies all white blood cells. CD45 is a surface adhesion molecule. And then we have to complex it to add another layer of specificity. We look at CD38, which is a pan-lymphocyte marker, which is expressed on B cells, T cells, NKs, and some other myeloid cells. But it's now a little more focused towards the myeloma, and it's generally highly expressed in myeloma too. And the third marker, which is the gold standard for detecting myeloma currently, is CD138, which is also another adhesion molecule, but it's specific, very specific for plasma cells. So if you notice, these are all cell adhesion molecules, and they are mediating the interaction between multiple myeloma and the tumor microenvironment. And we have been studying the cell-cell adhesion between myeloma and its tumor microenvironment intensively over the last 10 years. We now understand the cellular processes behind that and the cell signaling. And if we will have to go deeper in that, I'm more than happy to do so later, but it's not the main focus of this presentation. But what I wanted to emphasize that this interaction is very important for the development, progression, and dissemination of multiple myeloma. And now in our lab, we have characterized that very deeply, and we understand it very well. And what's the role of each of these molecules in the process of progression? One important feature of the multiple myeloma tumor microenvironment is hypoxia. We found that the more progressed is the tumor, there is more tumor hypoxia. Hypoxia is the lack of oxygen, and it happens because the progression rate of myeloma outrates the development of blood vessels. So we will have more tumor than what the body could supply blood vessels to it, so the tumors become hypoxic. Now, why is that important? We found that actually this changes dramatically the properties of the multiple myeloma cell. They become much more aggressive. They become much more metastatic. They start going to new places through egress from the primary site to the blood and homing to new sites. In addition, it makes it way much more drug resistant. The hypoxic cells are less going cell cycle, so they become more resistant to chemotherapy, and they display stem cell-like features, meaning they are dormant, but when given the opportunity, they will start growing so fast and making the relapse of the patient. But how is all that related? When we looked at the effect of hypoxia on the expression of CD138, which is the gold standard marker for detection of multiple myeloma, we found actually that this marker that has been used now for decades to detect multiple myeloma is lost from the surface of these cells. So we now know that there's a portion of multiple myeloma cells, which are hypoxic, we know they are there, that they are actually not displaying the main marker that people are looking for when we are looking for multiple myeloma cells. So we looked at the effect of hypoxia, and if you can see here, the effect on CD38 and 138 in the right box. And we found that while the expression of CD38 didn't change that much, the effect of CD138 went down dramatically, almost to less than half. So therefore, we learned that to detect multiple myeloma, we need to develop a strategy that's not it's not dependent on CD138. And why is that? Because it is specific for plasma cells, that's right, but it's variable expression. So you cannot rely on it to detect all the cells there. CD45 is also variable with hypoxia, also, although I didn't show it, but it is variable with hypoxia. It changes. And it's also widely expressed on all leukocytes. So it doesn't really give us any specificity. The only marker left is the CD38. However, it's not very specific to multiple myeloma. It's very highly expressed on myeloma cells. It doesn't change with hypoxia, but it's also expressed on other cells like B cells, T cells, NKs. Not to the same extent, not to the same level it is expressed in myeloma, but still it's present. So our idea was, let's use this kind of unspecific marker and make it more specific. How can we make it more specific? Simply by excluding all the others. For example, we will stain for CD38. We know we will be picking now all myeloma cells, the CD138 positive and the CD138 negative, which are in the black oval here. But we will also be picking other cells like T cells, B cells, NK cells, etc. So we will add antibodies to stain these specifically and exclude them from the total group. So we add CD3 to remove T cells. We add CD19 to take out B cells. To take NKs, we add CD16, and etc. So we have a line of exclusion markers, what we call CD3, CD14, CD16, 19, and 123. This will ensure, when we add them to the mix, we will ensure that we can exclude all the cells which are not myeloma, but still CD38 positive. Now, we put all these exclusion marker in one channel in the flow cytometry. For example, in the FITC channel, but it doesn't have to be. And we put the 38, which is the positive marker, on another channel, like the APC, but it doesn't also have to be. It can be any other color. So the idea here is that the multiple myeloma cell will be 38 positive, but negative for all the others, regardless if it's 138 positive or 138 negative. And I'll show you in a minute how this will actually start picking more myeloma cells, which the classic gold standard marker CD138 will not pick. What we started with here was that we combined normal bone marrow cells that they don't have myeloma. We combined these with a myeloma cell line. We added exactly 10% cells into the normal bone marrow cells. And once we added normal or normoxic myeloma cells, or we added myeloma cells which were put in hypoxia before the combination, then we tried to find them back, either with CD138, which is the gold standard, or with CD38 alone without the exclusion, or with the new method. So when we try to find with 138, we get in the normoxic cells, we get about 7% out of the 10. We get less than 5% if the cells were hypoxic, which actually means that half of the cells that we put, if they would be hypoxic, we wouldn't even detect them using the gold standard model. If we just use CD38 alone, we are picking now 25% instead of 10, because we are picking other cells, not only myeloma. But when we use the new method that we just defined, we pick back exactly 10%, which is what we put to start with. So we took that and we took it a little further in vivo, and we put myeloma cells GFP labeled, green fluorescence protein labeled, so we could track them. And we put them in mice, in skid mice, in different amounts. And we let them grow for almost 3 weeks. So we got mice with different levels of myeloma involvement in their bone marrow. we take the bone marrow out, just like we do in patients. We take a bone marrow biopsy or a bone marrow aspirate. So we isolated bone marrow from these mice, and we compared how much we can detect myeloma cells in the marrow using GFP, which is actually an artificial color that we put. So every cell that's GFP is by definition myeloma. Then we tried to see if we could find these using the gold standard marker. And as you see in the right part, despite that the amount of GFP/myeloma cells was increasing dramatically up to 65% in the bone marrow, the CD138 could barely detect 10%. So this is a huge false negative. While if we look at the new method, we picked exactly the same amount that the GFP found. This gives us another layer of confidence that now we are not missing any myeloma cell, but even more importantly, we are not picking any false positive. So we took that and we wanted to validate another layer in patients. So we took patient bone marrow aspirates, and we looked first of all at CD138 positive and negative. We just took the 138 negative portion, which is supposed to be no myeloma cells according to the classic definition. We detected with the new method. So we isolated the APC positive, FITC negative, as we defined it before. And we looked at the clonality. So myeloma cells, when there is myeloma, there's actually the cells detected there are always clonal. It means they either secrete kappa antibodies or lambda antibodies, because generally it starts from one clone. So that's another way to make sure that we have a clonal disease by identifying if it was kappa or lambda. And when we looked at, for example, this patient, we found that about 99% of the cells that we detected with the new model were all kappa. Now, in normal case, it will be about half and half. It can be a little more or a little less in a normal subject, but generally, the normal plasma cells distribution will be almost half and half. But when you find 99% of the cells kappa, which is one of them, it means that these are clonal, these are cancer. So we did that for a cohort of patients, of 20 patients, and we found that the marrow of these patients would include sometimes up to 35% cells, which are myeloma, according to our definition, but they are CD138 negative. And when we look at the clonality of these patients, actually they show significant clonality. It means they are not innocent plasma cells. These are clonal cancer plasma cells, which by the regular tests will be defined as normal cells or just missed. The regular classic model based on CD138 wouldn't even detect them. We looked, just to make sure that these cells are very clonal, we looked at how much non-clonal the percentile in these cells detected. We found that these all were less than 1% non-clonal. So all of these cells are highly clonal, meaning highly cancerous. Therefore, this tells us that we have developed now a new model that actually can detect a new subset of myeloma that was completely missed before. And if you look at these cells, these become very important in the definition of what we call minimal residual disease. So minimal residual disease refers to myeloma cells that are present in the bone marrow that after we treat them clinically, they stay there, they're almost undetected, and they induce remission. After remission, they induce relapse. So how that works, what's the concept? So if we treat and we bring the tumor burden, let's say 10 times lower, so we will have a kind of short period of remission. If we make the tumor burden 100 times lower, we will have a longer remission, and so on, until we get almost to no cells, which will say no remission. That's probably theoretical. Most of the time we get to like 10, 100 cells if we are really lucky. Then we have long periods of remission. So that these minimum amount of cells are very hard to detect. You know why? Because as I said before, most likely these cells are drug resistant, are the very hypoxic ones, which are actually CD138 negative. So it's now known that the definition of MRD will change how we treat, because it's not enough to just reduce the size of the tumor. We want to bring it to a level that's even these minimal residual disease will be gone. However, how to detect these with what's the optimal technology is exactly the question. So we suggested using this flow cytometry technique that we developed to detect minimal residual disease, because it's again independent of CD138. And we tried to find a connection between the amount of cells that we found in the bone marrow using our technology with the time takes the patient to relapse. So we took a group of patients. After treatment, they were defined cured. They were defined CR. And we looked into their bone marrow using the new technology or using CD138, the standard model. So with CD138, they all looked pretty clean, less than 0.5% if you look at the left box. And there is no difference. We couldn't really find any difference in the amount of CD138 cells in patients who relapsed fast, less than 24 months, or in patients who relapsed slow or longer. But when we look at the bone marrow with our new method, we found that those who relapsed fast actually have pretty high amount, more than 3% of the undetected cells using the classic techniques. Now we found about 3%, while the ones who relapsed late or had a slower relapse had less than 1% detected using the new method. So now we could correlate between the MRD, minimal residual disease, detected by our technology compared to the classic technology, and that we correlate with time to progression or relapse, while the classic MRD fails to do that. So in our case, for example, I would go and recommend the clinicians to continue the treatment for those who have a lot of myeloma cells left, like 3%, 2%, and to bring this even further down to deepen their response to therapy and make them relapse much later. From here, we wanted to go a little more exotic, I would say. And we thought, could we, instead of bone marrow biopsying patients, could we detect these cells in the blood? And currently, there is no way to correlate or to find almost any circulating myeloma cells, although in vivo models, we do see them, but we cannot find them in patients. And the reason is because the circulating cells, as we have shown, are very hypoxic. So most likely these do not have any CD38. And when we looked in animals, actually, we looked at GFP cells again, so we know these were circulating, but when we tried to find them with CD138, if you look at the right box, we found nothing. Because these circulating GFP cells, myeloma cells, are not expressing any 138. It's lost. But with the new model, we actually detected about 80%, I would say. In some of them, we detected almost 100% of the circulating cells. So with this method, we could somehow skip bone marrow biopsy, which is a very expensive and very painful procedure, by just taking blood from these patients and detecting with the right markers. We did that in patients, and we actually did find a lot of circulating cells in the patients, while with CD138, we found, again, nothing, less than 0.5%. So we tried also to look at if we can predict progression-free survival and see if that correlates. We did find a correlation, not as strong statistically as before, but it's statistically significant. It's highly statistical significance. So in patients who had fast relapse, we found high amount of circulating myeloma cells, while the ones who relapsed later, we found lower amounts of the circulating cells. So just in the end, to compare all the three technologies that we have available right now. So we have next-gen sequencing. It's quantitative and highly quantitative for detection of MRD. It is. While the CD138 is not that good for detection of 138, our new markers are highly quantitative. Prediction of progression-free survival, it is right with next-gen sequencing, not with flow cytometry based on CD138, but yes, for our markers. Time of analysis for next-gen sequencing, as we said, it's 1 to 2 weeks. In our case, in both flow techniques, it's 2 to 3 hours. Availability of the service for next-gen sequencing, we need to outsource. The flow is in every lab. And sample preparation, no primers. So we need primers and we need reference samples for next-gen sequencing. No such requirements are needed for flow. Detection of circulating tumor cells. It has never been shown yet by next-generation sequencing. I'm sure that at some point people will start exploring that. It's poor in CD138-based flow. We can detect it with our new technology using flow cytometry. So that's a huge advantage. And the cost is pretty low for both the flow techniques and the CD138. So I think the future is going to be something close to the technology that we developed. And at that point, I will thank you for listening, and I would like to open it for questions.
Thank you, Professor Azab, for that outstanding presentation. We will now move into the live Q&A portion of our webinar. Now, as a reminder to our audience, please submit your questions via the Q&A box. Okay, Professor Azab, here's our first question. Although CD138 is considered the gold standard marker for myeloma, as you mentioned, and others have also shown, that it is shed from the cell surface in some MM populations. Can you elaborate more on what the MM cells lose their CD138 surface expression?
Sure. As I mentioned before, CD138 is an adhesion molecule. It interacts with integrins to promote cell-cell adhesion. But we found that the expression of this surface marker is actually reduced due to many stresses. And probably tumor hypoxia is the most important one of them, but it's not the only one. For example, treatment with chemotherapy would even reduce it even lower. So if you think about these myeloma patients that were heavily treated with chemotherapy, and the cells are hypoxic and drug resistant and have CD138 low, now we treat them with more chemotherapy and they even lose even more the CD138 expression. Therefore, now these cells will become almost unseen in the patients. So we will observe the recurrence because of these cells, actually in most of 90% of the patients, because we miss these cells in more than 90% of the patients.
Thank you. Our next question. There have been several attempts to develop CD138 independent markers for myeloma. How is this new method different?
Right. Yeah, I agree. You know, especially there is a lot of research on that, especially coming from Europe. And the methods that are now used are really complex. They call them, as of their complexity, they call them next-gen flow cytometry, because it's based on 9 to 8 color flow, which makes it technically very complex and hard. You will need special machines to do that, special flow cytometry machines, and you will need very highly skilled technicians or workers to do it. Now, in research setting, this might work. In clinical setting, this will be very hard to implement. The great advantage of this technology that it relies on 2 colors. One color is positive, one color is negative. As simple as that. And as of instrumentation, actually these machines are available in every single hematology lab, in clinic or in research. You could even very easily do high throughput because it's simply just 2 colors, yes and no, and that's it. That's what I think is the huge advantage of the new technology.
Great, thank you for that explanation. Our next question, could you please elaborate more about replacing bone marrow biopsy with blood analysis for detection of MRD? This sounds like a winner, but seems like we're not quite there yet.
It is, I agree. We're starting. The point is that for a long time, you know, our group and many other groups, we were having very hard time finding any circulating myeloma cells at all. And now we know the reason why. Because these cells are hypoxic, they just lose their CD138 on the way to the new metastatic site. So with this new technology, now at least we can see them. Now, if this is going to replace bone marrow biopsy or not, that's definitely a bigger question. We might need much more research to prove that, especially in the MRD setting, where we know that just a little bit of the myeloma cells actually circulate, even when it's a full-blown disease. So in MRD settings, it's probably hard to rely on blood samples, but so we will have to still go to bone marrow biopsies. But for, I would say, routine detection and routine looking at tumor progression or just diagnosis of the disease, I would feel very comfortable using the circulating technology.
Okay, well, thank you. And we have time for one more question. Technically speaking, how do you see these markers being marketed?
Okay, so that's a good question, actually. So the idea here is that we take these markers as a kit. It's actually a bunch of antibodies that we put them all together in a vial. So the way we use it right now, we very much use this technology to detect myeloma cells in 3D cultures and in vivo. And the reason is that in 3D cultures and in vivo, they develop hypoxic conditions, and a lot of these cells really lose their CD138. So to go and detect them with the classic models, it's useless. So what we do actually is we have a vial that we mix all the antibodies together, and we just take the same amount and add it to the different myeloma samples. So to answer your question directly, I would think of it as a kit that has a mix of antibodies that will be sold as a vial that the researcher would take a certain amount per certain amount of cells that you want to detect myeloma in, and just analyze it directly by flow. I would even call it multiple myeloma detection kit.
Excellent.
Very easy to upscale.
It seems so. Well, Professor Kareem Azab, thank you so much for your time today and for your research. And we want to thank our audience too for their outstanding questions. We hope they found today's presentation to be informational and insightful. Now, today's presentation will be available for on-demand viewing. And to our audience, don't miss out on the other valuable presentations on our agenda. You can visit that agenda tab in the auditorium for a full listing. Thank you again for your participation. And until next time, goodbye.